Good afternoon. Okay, welcome to today's session. So today we are going to look at muscle tissue. Muscle tissue. So muscle tissue is one of the four basic tissue types, and it is the one that is whose function is for movement. It has contractile properties, meaning it can contract and relax. It can contract and relax. Okay, the muscle tissue has microfilaments inside the cells. Like I said, microfilaments when we were learning the cell, I talked about there is what we call cytoskeleton, the skeleton of the cell. And we talked about microtubules, we talked about microfilaments, okay? And so when you're talking about muscle tissue, you're going to find microfilaments in muscle tissue. And those microfilaments are the ones that are responsible for causing movement, okay? For causing the movement to contract and to relax because of a certain mechanism that we will look at. Okay, so that is that is how microfilaments work. Now, muscle tissue originates from the mesoderm. During embryonic development, we have the embryonic disc, and one of the layers of the embryonic disc is the mesoderm, and that is where muscle tissue originates from. Some functions of muscle tissue include one, movement. Obviously, you know that for you to move from one place to another, it is made possible by your muscles. The muscles have to move in accordance to how you want to move. For example, if you want to fold your leg, if your leg has to flex, it means that you have to your muscles, the thigh muscles have to contract. If your leg needs to relax or extend, it means the muscles behind your thigh have to relax, okay? So that is how muscle tissue helps in movement. Movement also of blood. You are going to find that in muscle tissue helps blood to move along the blood vessels. Blood vessels have muscle tissue around them, and this muscle tissue helps with movement of blood. Not only that, even the actual skeletal muscle, it will help the movement of blood, especially in the veins. The blood in the veins will move at a very low pressure because one, it is moving against against gravity, okay? It's moving one against gravity, and two, it is moving at a low pressure, okay? So you find that muscles in the legs are going to be helping together with the valves of the veins. They're going to be helping to squeeze the veins in order to allow blood not to backflow, okay? Including other things such as food. Food is going to be helped to move along the GIT by the muscles. It is because muscles initiate peristalsis that food is able to move along the GIT. So all those things are moved along in the body by the function of muscles, the function of movement. Then we have the second function, which is storage. Muscles store things like proteins. It is muscles that store things like energy units, okay? You are going to find energy in muscles. You're going to find protein in muscles. They help to store all these nutrients. The third function is for protection. Muscles help to protect internal organs. For example, the muscles of the abdomen, the abdominal wall muscles, they help to protect the organs that are in the abdominal cavity. So some of the muscles of the abdominal wall, they help to protect They help to protect the contents of the abdominal cavity. The other function is thermoregulation. Thermoregulation simply means to maintain temperature, the temperature of a body. So when you are too cold, the muscles will start shivering. When you start shivering, the muscles will start contracting and relaxation at a very fast rate. The contraction and relaxation of the muscle tissue, which will allow for you to shiver in order to generate heat. So that is what we are talking about when we talk about the functions of muscle tissue. Now, we are going to look at the classifications of muscle tissue. Muscle tissue is classified, one, according to microscopic appearance of the muscle tissue, two, according to the location where it is located, and the three, according to the function. Okay, so you are going to classify muscle tissue according to microscopic appearance, two, according to the location of the muscle tissue, and three, according to what function this muscle tissue actually performs. So the first, under microscopic appearance, we have two types. We have striated, okay, and then we have smooth muscle. Under microscopic appearance, there will be some muscles that when you look at it under the microscope, it will look like it has some stripes, and those are striated muscles. And then we have other muscles that will not appear to have stripes, and those are known as the smooth muscle tissue. Now, when it comes to classification according to location, we have one, the muscle tissue that is found on the body wall, meaning attached to the skeleton, okay, attached to the skeleton, and those are called somatic muscles or skeletal muscle, okay. And then we have the second type, which is located in the internal organs of the body. Those that are located in the internal organs of the body, and those are called visceral muscles. Now, those visceral muscles are called visceral because the organs that they are, the organs that they line are visceral organs. So all those organs that are inside your body are known as viscera. So the muscles that are associated with the viscera are therefore known as the visceral muscles, okay? The organs that are associated with the viscera are therefore known as visceral muscles. And then we have a third, which is the cardiac tissue. The cardiac muscle. Cardiac muscle, just like it sounds, is found in the heart. Cardiac muscle is found in the heart. And then according to function, muscles can be either voluntary or involuntary, meaning some muscles can work when you tell them to work. For example, if you want to move your leg, you're going to just move it because you want to. The legs will not move on their own. You have to want it to move, and then you tell it to move. However, there are some other muscles that will only work on their own without you telling them. For example, when you swallow the food, once it passes the pharynx, there is no way of stopping it unless you are sick. It's going to go all the way down because the muscles will immediately initiate peristalsis. So that is what that means. Or the cardiac muscles, for example. Okay. Now, when you are talking about cardiac, the types of muscles, this brings us to a broad classification. This brings us to three broad classifications. Okay, this brings us to three broad classifications. So 1, we are talking about skeletal muscle. 2, cardiac muscle. 3, smooth muscle. Now, skeletal muscle is striated. That is according to the microscopic appearance. Skeletal muscle is striated. Okay, according to location, it is somatic, meaning it is found on the body wall, and it is voluntary, meaning you can use it as you please. And then we have the second class, which is the cardiac muscle. Cardiac muscle is also striated, all right? It is involuntary, meaning you cannot use it as you please. It works on its own. You don't have to tell it to pump, all right? And then we have smooth muscle. Smooth muscle is not striated, okay? It is visceral, it is found on internal body organs, and it is involuntary. Those are the 3 broad classifications of muscle. Now, when you are talking about muscle tissue, the cells of the muscle tissue have specific functions. They have specific words that we are going to be using when we talk about muscle tissue. One of the specific words is that when you're talking about a muscle cell, you can call it a muscle fiber. One muscle cell is referred to as a muscle fiber. The cytoplasm of muscle tissue is called sarcoplasm. The cytoplasm of a muscle tissue is called sarcoplasm. The plasma membrane of muscle fibers is known as the plasma membrane, which is the cell membrane of a muscle fiber, is known as sarcolemma. The smooth endoplasmic reticulum of muscle fibers is known as the sarcoplasmic reticulum. Sarcoplasmic reticulum. So those are the 3. Those are some of the words that you're going to be using when you're talking about muscle tissue. Now, when you talk about the classifications, these are the pictures that are going to show you the 3 classifications of muscle tissue. So you have 1 here, this is the skeletal muscle. 2, we have cardiac muscle. 3, we have smooth muscle. Now, in this picture, you will realize that when you look at skeletal muscle nicely, you will notice that 1, it appears as if it has stripes, okay? Long across, like it has stripes. This is why it is known as a striated muscle, because when you look at it under the microscope, you are going to see these stripes, okay? These stripes running from this side to this side, from this side to this side. So now we are talking about the properties of each of these muscle cells. We are talking about the properties of each of these muscle cells. So 1 is the fact that skeletal muscle is striated, okay? The second property is that skeletal muscle resembles a rod or a tube, okay? So they are rod-shaped. So they'll look like a rod. They are elongated like that, elongated, okay? So they will look like a rod or a tube. The third property is that skeletal muscle will have more than 1 nucleus. A skeletal muscle fiber will have more than 1 nucleus. So that is what you should take note of. 1 it has stripes, meaning it's striated. 2, the shape is elongated, it looks like a rod. 3, the skeletal muscle fiber will have more than one nucleus. And 4, the nuclei will be peripherally located just under the sarcolemma, just under the sarcolemma. Those are some of the properties of skeletal muscle. The second muscle type that we're looking at here is this one in here. This is the cardiac muscle. Cardiac muscle properties include 1, it is striated. When you look at it, you're going to see these lines still. So it is striated. 2, it is branched. You will notice that it looks like branches, like they are branching from one another, okay? From one another. 3, you will notice that each fiber has one nucleus. Each fiber has one nucleus, okay? So you will notice that each fiber has only one nucleus. It is not multinucleated. The third property, you will notice that the cardiac muscle will show these lines when you are looking at it under a microscope. These lines are where one muscle fiber starts and another muscle fiber ends. So if this muscle fiber is coming from this side, it will come and it will end here, and another muscle fiber will start and it will end here, just like that. It will, so this can be one muscle fiber, okay? This is one muscle fiber, this is one muscle fiber, this is one muscle fiber, but along the way, you will see that they appear branched. Now, at the point where each muscle fiber is ending, there is going to appear a line, and this line is known as an intercalated disc. And this line is known as an intercalated disc. So you are going to find that 1, the cardiac muscle is striated. 2, the cardiac muscle fibers are branched. 3, the nuclei are, each muscle fiber has only one nucleus. 4, you are going to find intercalated discs in cardiac muscle fiber, okay? You are going to find intercalated discs in cardiac muscle fiber. When you go to smooth muscle, on the other hand, you will not see these striations. So smooth muscle is not striated. That is why it is called smooth. You are only going to see that it is smooth muscle. It's not going to show any striated. And so it is smooth. It is not striated. The other thing you will notice is that it is spindle shaped. So if you look at this muscle, it is spindle shaped, sharper in the ends. So it will appear like a spindle shaped, okay? It will appear like spindle shaped, thicker in the middle and sharper and smaller in the sides. So it is not striated, it is spindle shaped. It is uninucleated, so each fiber has only one nucleus, and that nucleus is located in the center. So it is centrally, the nucleus is centrally located. That is what that means, okay? So now, let us look at striated muscle. Striated muscle, or let us look at skeletal muscle, not striated muscle. Let us look at skeletal muscle. Skeletal muscle is the majority of the muscle tissue in the body. If you think about it, the muscle which attaches to your bones is the majority. Like I was saying, it contains parallel bundles, which are long, cylindrical, multinucleated with transverse stripes. Okay, the contraction is quick, powerful under voluntary control, innervated by somatic and branchial motor nerves. Okay, we're going to look at the motor nerves and you're going to be connecting very soon. So this is how skeletal muscle is going to appear. It will look like it will look like a rod. It is rod shaped. Okay, this is how it develops all the way from there, and then at the end of the day it will look like this. Then it is a long cell. Okay, a very long cell. So the functions: voluntary movement, for example, your sphincters. You don't want to go to the toilet, you're going to hold it unless you are sick. Okay. Another function is locomotion, movement from one place to another. Posture, the way you are able to sit like Leo in class and not like Jumbo. Okay. Position, poise, heat production. So we have talked about all these things. Okay, so those are the functions of skeletal muscle. Even these we have talked about. These we've talked about the histological features of skeletal muscle. We said cylindrical, striated, long cells, elongated, peripheral, nuclei, just under the sarcolemma, and multinucleated. Okay. Now, the arrangement of skeletal muscle is arranged in such a way that these bundles, okay, these bundles will be arranged in this way. You are going to have muscle fibers, millions of muscle fibers, okay? Now, one muscle fiber has, remember I talked about microfilaments. One muscle fiber has microfilaments, those cytoskeletons in the cells. They are nicely arranged in skeletal muscle. So the microfilament will run from, if this skeletal muscle is this long, it means that microfilament will run from one end to the other end. They'll be connected, interconnected microfilaments, which will appear to be one line all the way from end to end. Okay, very nicely located, and they pack the muscle fiber. They completely pack the muscle fiber such that the cytoplasm is just around those microfilaments, and then we have nuclei which will be pushed towards the peripherals, towards the peripheries, and the arrangement is nicely done in muscle fiber. Okay, so this picture is showing us the arrangement of muscle tissue, showing us the muscle tissue. If you look at this thing, this is a bone. Okay, for example, in fact, this is the humerus. This is the humerus. Okay, so let's get, for example, this. So this means this muscle, if this is the humerus, and looking at where it's passing, we are going to say this is the biceps. Okay, these are bicep muscles. Now, the way these biceps, you know those biceps muscles that you guys go and you lift up weights and then you build up. This is the biceps. Now, they have gotten this biceps muscle and they have cut it in the transverse section. They have cut it and they have pulled some out, and then they have gone ahead and put one muscle fiber. Remember I said that one muscle fiber is a cell, one muscle cell. They have pulled it out, and they have also cut it in the transverse section. And once they cut it in the transverse section, you'll be able to see those dots there. Those are the microfilaments that we're talking about. And in the peripheries, we are able to see the nuclei. Okay, we are able to see the microfilaments and the nuclei in this muscle fiber. Now, what happens is this microfilament, this muscle fiber, is surrounded by a thin layer of connective tissue. So all muscle fibers are surrounded by a thin layer of connective tissue. That thin layer of connective tissue is known as endomysium. Okay, that thin layer of connective tissue is known as the endomysium. And then when you get several of these muscle fibers, so this is a muscle fiber which has been pulled into the front. This is another muscle fiber. This is another muscle fiber. So these are cells. This is one cell. This is another cell. This is another cell. This is another cell. So these are muscle fibers, muscle cells, one cell, two cells. So these are millions of muscle cells. Okay, now each of them is covered by this endomysium, which is a thin layer of connective tissue. When you get these cells together with the endomysium, you put them together to form one bundle. This bundle is known as a fascicle. This whole bundle here, the bundle that contains muscle fibers, is known as a fascicle. This bundle is also surrounded by a layer of connective tissue. This bundle is also covered, surrounded by a layer of connective tissue. And this connective tissue is known as perimysium. Okay, this connective tissue is known as perimysium. In this connective tissue, we have enclosed in it blood vessels, lymphatic vessels, and nerves. All right, so those blue ones are veins, those red ones are arteries, and then we have nerves and lymphatic tissues, which you cannot see here, lymphatic vessels, which you cannot see here. Now, when you get this fascicle and you combine it with other fascicles, okay, this fascicle, and you get another fascicle and another fascicle and another fascicle, and you put them together. And then around it, there is going to be another layer of connective tissue. There's going to be another layer of connective tissue. That connective tissue, which is the most external, is known as the epimysium. And this epimysium, once it covers everything, now this is the skeletal muscle. And you have your biceps, okay? This is the bicep. These are your biceps muscles. So that is the arrangement of the muscle tissue. Okay, that is the arrangement of your muscle tissue. Okay, so those microfilaments inside the muscle tissue are known as myofibrils. Those are longitudinal, long cylindrical filament bundles that mainly fill the sarcoplasm. Vast mitochondria and sarcoplasmic reticulum lie between the myofibrils, okay? The myofibrils are composed of myofilaments, muscle filaments, those myofilaments, okay? That is what that means. Now, the organization within the muscle fiber now. Let us look at the organization of these filaments within the muscle fibers. Let us look at the organization within the muscle filaments. If you look at this diagram here, this diagram is nicely depicting how a muscle is. Remember that diagram we were looking at? This one. Those biceps we were looking at, it is this one. So from these biceps, we have decided to pull out one muscle fiber, meaning one cell. We have decided to pull out this one muscle fiber, and we have decided to enlarge it. Okay, we have decided to enlarge it so that we see the arrangement of those myofilaments inside. Okay, myofilaments are the cytoskeleton of muscle fiber. They are protein in nature. They are the ones that allow for the contraction and relaxation, okay, of the muscle tissue. They are the ones that are the functional units of muscle tissue. Like I said, they are protein in nature, okay? They are protein in nature. They are arranged in such a way that they are made up of proteins known as myosin, and they are made up of proteins known as myosin and actin. They are made up of proteins known as myosin and actin. Now, when you look at a muscle fiber, like I said, those small protein filaments, they are arranged in such a way that they will form a straight line. It won't just be one, there will be several of them, millions of them. They'll move from one end of the cell to the other. So this is the road that it has made. Now, the arrangement is in such a way that they are organized in units known as sarcomeres. They are arranged in units known as sarcomeres. And these sarcomeres, they are the ones that aid in the contraction of the muscle tissue. So for example, a sarcomere starts from one Z line to another Z line. So if you look at this diagram nicely, you are going to see that there are different shades. The same way that these ones are appearing here is the same way you are likely to see this thing under a microscope. This is an actual electron micrograph, an actual light micrograph, okay? This is exactly what you're going to see under a light microscopy. You are going to see one, there will be this dark line and another dark line, and then you are going to see a darkened area here, okay? Then you're going to see a line in between the darkened area, and there will be this line that will be slightly dark, but not very dark. This is because this area is able to attract more light, whilst this area is unable to attract more to attract light so much, and this area just like that. Okay, so that is what they are. Now, this area which is bright, which is light, is known as the I band. This area is known as the A band. Okay, this area is known as the I band. Now, a sarcomere will run from this line, this dark line, to this dark line. The arrangement here is such that we have, like I said, two types of protein. We have actin. Those are thin filaments. Those are the ones that are in this light band. And they go all the way from here to somewhere in the middle of this dark line, this dark area. And even this side, we were going to have actin, those thin filaments which will go from this dark area to this. So from this Z disc to this Z disc, this is what is going to be called the sarcomere. These are the contractile units. Another sarcomere will start from here to another Z line, just like that, until the end of this muscle fiber. Now, this arrangement is in such a way that when you look at this picture here now at the bottom, you are going to see that this thin filament, this is what I was saying, this thin filament is running from this Z disc all the way up to somewhere in the middle of the darkened area. This darkened area is darkened, it has got thick filaments, okay? Thick filaments that are myosin, okay? These are actin. And so The thin filaments will move all the way from one Z disc to there, about in the middle, but where exactly they end will depend on whether the muscle is contracted or whether the muscle is relaxed. And so we are going to have this actin, these actin filaments here, and then we are going to have the myosin filaments there, and then we are going to have the actin filaments just like that, and then the Z disc and another sarcomere on the other side, completely until the end of this muscle fiber. In the middle of the A band, we see another line, okay? This other line is known as the M line. We have the M line. And then when I was telling you about this one, I told you that we have this area which is slightly light, not very light, but slightly light. It is known as the H zone. Here you find only thick filament. Here you find thick filaments and thin filaments. Here you find only thin filaments, okay? So that is what it is. This is the M line, this is the H zone, this is the thick, here you find thick and thin filaments, here you find thin filaments only, and here you find thick filaments only. Okay, so that is the arrangement. So if you look at now this drawing, it is showing that this is one microfilament inside a muscle fiber, and this microfilament is running from one Z disc to another Z disc, and it is showing you the I band, which is the light area where we have the thin filaments alone, and the dark area, which is the A band, okay? And then we have the H zone in the middle of the A band, and we have an M line, okay? So you have to know all these. The other thing you have to know is when you cut, this is, by the way, this is one sarcomere up to here. From this Z disc to another Z disc, this is one sarcomere. This is another sarcomere. From this Z disc to the next Z disc, that is another sarcomere. From this Z disc to another Z disc, that is another sarcomere. From this Z disc to another Z disc, that is another sarcomere. Now, when you cut these sarcomeres in the transverse section, whatever is there is what you'll be able to see in these circles that are showing you here. So for example, if you cut this sarcomere at the light band here, at the I band here where there is just the thin filament, okay? The question is, what are you able to see? This is what you're going to be able to see. There is these other thin proteins here known as titin, okay? You'll also be able to see them there. Those titins are coils. They are coiled in nature, so they just help to give the muscle tensile strength in coiling and recoiling. So you have this. Now, when you cut this one sarcomere from the other, dividing the sarcomeres at the Z disc, okay, at the Z disc, this is what you're likely to see. You're likely to see, you've seen there is titin here, okay? Then you're likely to see the connection of the filaments, the connecting filaments, all right, here. So this is what you're going to see. If you cut this sarcomere at the A band, okay, at the M line, within the A band, you cut it here, okay? Remember we said that here at the M line, you only have thick filaments alone. And so those are the ones you're going to see there, thick filaments alone. So just like that, when you cut this one at the H zone, okay, you cut it at the H zone, you're also going to see these thick filaments. And when you cut it at the A band where there's a mixture of thick and thin filaments, you're going to see thick filaments and thin filaments over there. So that is what the arrangement within the muscle cell looks like. Okay, so I've explained the muscle, the myofilaments. I've talked to you about them. Now, when you look at the thick filaments, which I already told you are myosin in nature, they are made up of proteins known as myosin. The thick filaments have heads and tails. Okay, the thick filaments have heads and tails. So when you look at this thick filament, okay, it is made up of these smaller filaments banded together, okay? So these smaller filaments banded together are myosin molecules. This myosin molecule itself, only one of these is now taken from here and magnified for you to see the tails here and the heads there. Okay, these are the thick filaments. Now, they are made in such a way that the heads have a binding site for actin. Remember, we said that actin is what makes up the thin filaments. So the thick filaments, the heads have a place where there is a binding site for actin. Okay, that is what that means. And then we have the thin filaments here. These thin filaments are made up of three more important molecules for you to take note of. One, they are made up of troponin, okay? This triple complex here. And the troponin is where they have myosin binding site. On top of the troponin, you have a site where this myosin head is going to bind. On the other hand, here, this actin will bind on that actin binding site, okay? The other thing that makes up the other thing, sorry, here, this is the calcium binding site. The myosin binding site are on the F-actin here. F-actins are they are made up of these circles here, these tiny beads. Each tiny bead is known as a G-actin. Each tiny bead is known as a G-actin. When you get these beads together and you make a chain, they are known as F-actin. On each G-actin, you're going to find myosin binding sites. For that to happen, there should be calcium that should be produced to come and bind to troponin. I will say that again. We have myosin, which are the thick filaments. This myosin is made up of a lot of myosin molecules banded together to form thick filaments. The one myosin molecule has heads and tails. On the heads, we have a spot where there will be a binding site for actin. We said that the thin filaments are the ones that are made up of actin, a protein known as actin. On the protein known as actin, we have three things that I need you to take note of. One, we have troponin. Two, we have F-actin. Three, we have tropomyosin. One troponin has a point on top where calcium is supposed to bind in order for this for muscle contraction to occur, okay? In order for muscle contraction to occur, we have a calcium binding site. Under the troponin, we have this tiny chain here. It is tropomyosin. And then we have another chain made up of these big beads, and these beads are known as G-actin. One bead is G-actin. When you make a chain of these beads and they are here arranged like that helically, these are now called F-actin. That is one F-actin. So on each G-actin, you have a myosin binding site. Remember, the myosin is this one. So the myosin is going to be binding on some of these. In fact, not each G-actin has myosin binding site. I think about 2 or 3 after the troponin will have myosin binding sites. So that is what that means. You have myosin binding sites on G-actin, and you have actin binding sites on the heads of the myosin. Okay. So these are the explanation. We have F-actin, double helix strands, okay, made up of G-actin monomers, those beads I was telling you about, okay. We have tropomyosin, coil of 2 polypeptide chains found in grooves among the 2 actin strands, okay, among the 2 actin strands, okay. These are the 2 strands of actin, the beads, the G-actin. So you find, that is where you're going to find tropomyosin. And then we have troponin. We have troponin 3 subunits. Okay, we have one that has a binding site for calcium. We have another that binds to tropomyosin. Okay, and we have another which regulates the actin-myosin interaction. Okay, we have another which regulates the troponin action interaction. Okay. Now, we have other accessory filaments. Like I was telling you, when you look at this other diagram, we have another type of protein here, the green ones here. These are titin. They are known as titin. They are made up of proteins known as titin, and they are accessory proteins. Then we have nebulin, which binds and anchors thin myofilaments laterally. It binds and anchors thin myofilaments laterally. Okay. So those are the accessory proteins. Like I was saying, we have A bands with thick filaments and overlapping portions of thin filaments, okay? H zone, all these things I think I have already talked about. Sarcoplasmic reticulum is specialized for calcium sequestration. Sarcoplasmic reticulum is specialized for calcium sequestration. Depolarization, you're going to learn about depolarization, membrane potentials. You're going to learn about that in physiology. So you're going to understand that very much when you do the membrane potentials and depolarization and the propagation of impulses. Okay, depolarization of the sarcoplasmic reticulum membrane, that is the smooth endoplasmic reticulum, which causes release of calcium, is initiated at specialized motor nerve synapses on the sarcolemma. I think these you're going to go into detail in physiology. Okay, you're going to go in detail in physiology, but I'm going to explain to you how muscle contracts. I think that there is some along the knots. So mechanism of contraction. Calcium binds to troponin. Remember, we said that on troponin we have calcium binding sites. So calcium will bind to troponin, and this will cause tropomyosin to change shape. Okay, to change shape and allow the myosin heads to bind to actin subunits. So when you look at this, okay, you have calcium coming in to bind to troponin. And once that happens, those green bands there, they're going to change shape. They're going to change shape and they're going to allow for the G-actin, which have myosin binding sites, to bind to the myosin, okay? They're going to allow for the myosin binding site to bind to the myosin. The myosin heads then pivot with ATP hydrolysis, which pulls the thin filaments along the thick filaments. This is much easier when you're looking at a video, so you can try to check for a small sum to my videos and see how this looks. Because when they bind, so if you look at this is in the process of binding. This is binding, and then when they bind, they're going to kind of pull, okay? They're going to kind of pull the thin filaments such that the thin filaments will move towards the middle, okay, in such a way that the thin filaments will move towards the middle, and that means the muscle has contracted, meaning it becomes shorter. So this is happening at different points in that muscle fiber. Remember, the sarcomere are a lot of them on one muscle fiber. So we have this sarcomere and the next sarcomere. So this is happening on all the sarcomeres. And so you find that once this actin filament has been pulled towards the middle, it means the muscle has done what? Has contracted. Okay, it will be pulled. So you've seen these thin filaments have been pulled in such a way that they've come to the middle and they've actually even overlapped, and therefore you have flexed your elbow joint, because it means that the biceps have what? The biceps have contracted. That is what that means, okay? On the other hand, a relaxed muscle means that these now they go into place. The myosin and the actin are no longer interacting, and so they go back to their normal position. Okay. This is just a picture of a motor end plate. This is a nerve. Okay, this is a nerve, and this is an axon of a neuron, which gives out these projections at the end, and these are terminal buttons, and they're bringing impulses to come and move, because for the muscle to move, it has to work hand in hand with the nervous system. Okay, so this is the same thing that is showing. It's just showing how the muscle tissue is depolarized. All those things you will learn in detail when you do membrane potentials in physiology. So this is a neuron, and it is bringing the neurotransmitters and allowing for depolarization of the sarcolemma, okay? And so there'll be release of calcium, and all those physiological things are going to take place now in the muscle tissue. This is an actual electron micrograph. This is a drawing, but this is an actual electron micrograph, which is showing us this neuron, which is bringing the impulse, and these are motor end plates. Motor end plates are a place where there is interaction between a terminal button of a neuron and a muscle fiber. If you look at these muscle fibers, they look striped. They look like they're striated. So these, and they look like they're rod-shaped. So we can safely say these are skeletal muscles that are being depolarized, okay, or that are being innervated right now, so that this person can move the hand, okay? So that is about skeletal muscle tissue. The second type of muscle is cardiac muscle. We have talked about that it is striated, branched, the intercalated disc, there is involuntary contraction, and cannot divide. There are no satellite cells. Satellite cells are supportive cells, okay, and it does not regenerate. It is the heart's pumping unit, okay? So we have said that this one originates also from the mesoderm, okay? The cell morphology, shape is rectangular in nature, okay? One fiber is rectangular in nature, okay? They have intercalated discs between each of them, okay? And those are the things that are going to make you distinguish one cell from the other. So this is cardiac muscle fiber. This muscle fiber is starting from here, okay? And it has been cut in the middle, and it is running all the way up to here, where if there was another one connected here, you would have seen an intercalated disc. This is an intercalated disc. The muscle fibers appear branched, okay? Each muscle fiber has one nucleus, okay? You're going to find one nucleus in each muscle fiber. Sometimes it can be two if there is a branch, like this one has a branch. Sometimes it can only be one in the middle there, okay? So those are ways in which you can tell. One, it is striated also. Two, it has intercalated disc. Three, it is branched. Four, it is uninucleated. Only binucleated if maybe the nucleus is found in one branch, okay? So that is, those are some of the features of cardiac muscle fiber. This is an actual electron micrograph of cardiac muscle. It has been cut in a longitudinal section, and you are able to see that there is this one cardiac muscle fiber, and these are the nuclei that are found. This is a drawing, okay? These are intercalated discs where one muscle fiber is ending and another muscle fiber is starting. You can see that there are branches here. This one is branched. This one is branched here. You can see that there are branches on this cardiac muscle fiber. And this is another light microscopic diagram, like actual light microscopy that is showing us cardiac muscle. You can tell that one, there is an intercalated disc here and another intercalated disc here. You can tell that there is one nucleus here. You can tell that some of these cells or fibers look branched like this one. There seems to be a branch somewhere here. There's an intercalated disc here. Something also that looks like an intercalated disc there. It also looks like it is striated. It doesn't look smooth. So those are the features of cardiac muscle. Okay. Okay. Transverse regions of each disc have desmosomes and fascia adherentes. So those are the ones that help the cells to hold on to each other. Okay, those ones bind cardiac muscle cells firmly together to prevent pulling apart under constant contractile activity. So you have desmosomes, okay, and fascia adherentes, which resemble the zonular adherents, okay? So we have fascia adherentes. Let's see. Okay, so we have these desmosomes here between between one cell and another cell. Along the intercalated discs, we have these adhering junctions which help for muscle cells, for these muscle fibers to hold on to each other. And then we have these gap junctions which allow for the movement of molecules from one cell to the other, okay, which allow for the movement of molecules from one cell to the other. Okay. So like we said, each cardiac cell possesses one or two nuclei centrally located, which stand pale. Surrounding the muscle cells is a delicate sheath of endomysium, the same arrangement that we talked about. There is the endomysium, there is perimysium, there is the epimysium. Sarcoplasm is cytoplasm. There's numerous mitochondria for energy provision, numerous myofibrils. There is also sarcoplasmic reticulum. Those are things that we have talked about. The other thing that you're going to notice in cardiac muscle are stored triglycerides, seen as cytoplasmic lipid droplets. Okay, there will be glycogen granules also. Okay, there will be a pigment which is known as lipofuscin. Then you are going to have T-tubules, plasma membrane invaginations, more numerous and larger. Okay, those T-tubules, don't worry about those in anatomy. You are going to learn in detail when you look at physiology and membrane potentials. Okay. So muscle of the ventricles is much thicker than that of the atria. Atrial muscle cells are somewhat smaller with markedly fewer T-tubules. Membrane-bound cytoplasmic granules found near the atrial muscle nuclei release the hormone atrial natriuretic factor that acts on kidneys to affect sodium and water excretion. Okay, so those are just points of addition, okay? So the contraction is intrinsic, meaning it only happens right there in the heart and it is spontaneous. You don't have to tell it. It will happen whether you like it or not. Impulses for the rhythmic contraction are initiated, regulated, and coordinated locally by nodes of specialized myocardial fibers. Those nodes, the atrioventricular node, the sinoatrial nodes, all those nodes, they're the ones that carry out the function of initiation of the contraction. The third muscle type we're going to look at is smooth muscle. Smooth muscle is also known as visceral muscle, like I said, because it is usually found in visceral organs, the organs of the internal, of the insides. Intermediate abundant muscle type, meaning the most abundant was skeletal. The second abundant is smooth muscle. It is involuntary attached to tubular, hollow cavities or sinuses. For example, tubular, for example, the GIT, for example, the blood vessels. Okay, these support endothelial and epithelial linings. Endothelial linings in blood vessels, epithelial linings in organs such as the GIT. So you're going to find those there. The functions of smooth muscle include 1, peristalsis, 2, regulation of blood vessel diameter. It is the smooth muscle which is responsible to increase the diameter or reduce the diameter by contracting, okay? They are also responsible for regulating bronchial diameter, regulating pupil diameter. So they are also found in your iris, okay? So they are also found in the iris. So you are going to find smooth muscles in those internal organs. They are elongated, they are tapering, meaning at the ends they appear thin. They are not striated. They have a single long centrally located nucleus in the middle of the cell okay, at the point where the cell is broadest, where the cell is biggest. So this is generally the general appearance of smooth muscle. You are going to have a spindle-shaped muscle, which is thickest in the middle. It is tapering towards the sides. This nucleus is elongated, found in the middle, okay? Found in the middle, okay? You are going to find that for this, it is not striated because the thick and thin filaments are arranged in a different way. So this is the arrangement of the thick and thin filaments, okay? The thick and thin filaments are arranged differently. They are arranged in a manner as if they are obliquely arranged, okay? They are obliquely arranged, and so even the way they are going to be working, well, it will be the same as the skeletal muscle, but the contraction will appear slightly different. The endometrium is a sheath of thin basal lamina and fine network of reticular fibers that surrounds each fiber. Okay. I don't know why this slide is found here, but that is talking about those those outer surroundings of the mass. When you look at smooth muscle under a microscope, this is what you're going to see. You're not going to see any particular arrangement. Okay, this is what you're likely to see. The only thing you're going to see, yes, is a fascicle, a group of muscles, and yes, there's connective tissue over there, okay, the perimysium, okay. Then you will notice that some of them, when they appear something like this, then they were cut in the longitudinal section, okay. Now, when you look at muscles of, for example, the GIT, we know that muscles of the GIT will have, some muscles will be longitudinally oriented, while other muscles will be circularly oriented. So for example, if you get the duodenum and you cut it in the transverse section, not in the longitudinal section, you cut it in the transverse section, you are going to see one layer which is longitudinally, whose cells are going to look like they are straight, and the other layer which cells are going to look like you've just cut it right in the middle, as if you've cut it, you've removed the top part from the bottom part. Now, because you have cut this organ in the transverse section, it means those ones that you have cut in the middle to remove the top part, it means they are longitudinally oriented when they are placed in a human being. It means you've cut them in the middle. And these ones that are going to look long, it means they are circularly oriented. They are circularly oriented. So this will be the internal circular layer, and this will be the external circular layer. Okay. So what we're saying is you are going to get this, if you get this duodenum, you cut it in the transverse section, you are going to notice that you're going to see the two layers, the circularly oriented layer and the longitudinally oriented layer. And this is the same thing that you're seeing on this other light microscopic diagram, where you're going to see other cells seem to be longitudinally oriented, whilst others seem to be circularly oriented. So that means that these ones, the ones that you just cut in the middle, you are only seeing one point, meaning it's the ones that are elongated. These are the ones that have been cut in transverse section, and these ones have been cut in longitudinal section. Okay, so that is what we are going to find. So these smooth muscles have mitochondria, polyribosomes, rough endoplasmic reticulum, Golgi apparatus, and all the Golgi apparatus will be located near near the nucleus. Okay. So the fibers have rudimentary sarcoplasmic reticulum, but they do not have T-tubules. Those T-tubules, those are invaginations on the sarcolemma. And like I said, T-tubules, you're going to know them very soon. You're going to know them very well once you learn that term. So their function is unnecessary in these smaller tapering cells. That is why you're not going to have T-tubules. The axon and myosin are organized differently from those in striated muscles, which we've already talked about. Okay, these thick and thin filaments are arranged in such a way that they crisscross, like I said, in an oblique manner. They'll crisscross in an oblique manner. You know, a crisscrossing, they'll crisscross each other in an oblique manner, which means even the contraction will be different. The way that they will contract will look different from skeletal muscle or from striated muscle. In this one, myosin filaments have a less regular arrangement among the thin filaments and fewer cross bridges than in striated muscle. Okay, so actin filaments lack troponin. Instead, they use calmodulin and calcium sensitive myosin light chain kinase in the contraction mechanism. Smooth muscle cells also have intermediate filaments composed of desmin. Okay, intermediate. So the take-home point is that the contraction of smooth muscle is different from the contraction of skeletal muscle. The contraction of smooth muscle is different from the contraction of skeletal muscle. You just for the sake of interest, you can go ahead and read about the, if you are interested to read about the muscle regeneration, you can go ahead and read about muscle regeneration. Otherwise, we end here for today. Okay, we end here for today and see you next time.